Scientists have built a computer model that can quickly scan the entire genome of cancer cells and identify mutations that occur more often than expected, showing that they encourage tumor growth. This finding, according to a new study by the Massachusetts Institute of Technology funded, in part, by the National Institutes of Health and the National Cancer Institute. Distinguishing this dangerous driver mutation from neutral passengers can help researchers identify better drug targets. To increase these efforts, the team led by MIT has built a new computer model that can quickly scan the entire genome of cancer cells and identify mutations that occur more often than expected, showing that they encourage tumor growth. This type of prediction is very challenging because some genome regions have a very high frequency of passenger mutations, sinking the actual driver signal. We created probabilistic and learning methods that allow us to get a very accurate model of the number of passenger mutations that should be anywhere in the genome, said Maxwell Sherman, postgraduate student MIT. Then we can see the entire genome for the area where you have an unexpected accumulation of mutations, which shows that it is the driver's mutation. In their new study, researchers found additional mutations in all genomes that seem to contribute to tumor growth in 5 to 10% of cancer patients. This finding can help doctors to identify drugs that will have a greater chance of successfully treating patients, the researchers said. At present, at least 30% of cancer patients do not have a detected driver mutation that can be used to guide treatment. Sherman, MIT Adam Yora postgraduate student, and former MIT Oliver Prebes assistant research assistant is the main author of this study, who appeared today in Natural Biotechnology. Bonnie Berger, professor of mathematics Simons in MIT, and head of computing and biology groups in Computer Science and Artificial Intelligence Laboratory, are senior writers of this study, along with Po Ayu, assistant professor at Harvard Medical School and Harvard associate members from Broad Institute of MIT and Harvard. Felix Dietlein, a professor at Harvard Medical School and Boston Children's Hospital, also a writer of this paper. New tool. Since the human genome was sorted 2 decades ago, researchers have explored the genome to T.